Good morning, everyone. Welcome to the Friday Coulter seminar series. It's my pleasure to introduce Dr. Venugopal, a professor in integrative biology at University of California. Although I'm not sure I'm the right person to be introducing her because I see that she was getting her degree at Ohio State, and I got my degree at University of Michigan, which makes us pretty fiercely competitive college football rivals, I believe. But nevertheless, her research is so engaging that I was willing to do this introduction. And one of the things I like about her portfolio is the way that she merges experiment and modeling, which is the philosophy I think all biomedical engineers should have, and certainly the one that we have in our lab. And she does this both in her research and in her community outreach, running workshops that merge science and computational mathematics. And her main research focus, which I think we'll hear about today, is going to be about how neural circuits are reconfigured by neurodegeneration. So we all know, presumably, that neurodegenerative diseases are a big problem, everything from Alzheimer's to ALS. But one of the questions that's still largely not understood, not answered, is how does the neural degeneration manifest into these deficits? And she does a lot of great work looking at how the neurodegeneration can reorganize things like microcircuits and how that ultimately manifests in changes in phenotype, changes in local behaviors. So without more ado, let's all listen to what she's got to say on this topic today. I know I'm very excited to hear. Thank you.
Thank you very much for the kind introduction, Dr. Dan Zeiger. I hope I'm pronouncing your last name correctly.
Yes, well, it's definitely more correct than the way I pronounced yours.
No, mine was completely fine. Thank you for inviting me, Dr. Chung, and this is such a great opportunity for me to be here. And like Dr. Dan Zeiger suggested, he beautifully described my work. I think I don't even need to say anything extra as an introduction. So I really appreciate the introduction. And I'm really happy to be here because this is such an interdisciplinary group and such an apt group to share my work. Without further ado, I'm going to share my screen and have you all hopefully enjoy the talk. So thank you all for coming once again. And I hope you can all see my screen. Could you all give me a thumbs up? Let me see here.
Yes.
Okay, great. Thank you so much. So as the title of my talk goes, I'm going to be talking about modeling neurodegenerative disease dynamics. We are looking at neural microcircuits and how they are reorganized as the disease progresses, especially at the pre-symptomatic stages. So my talk will basically address the neurodegenerative disease called amyotrophic lateral sclerosis. So this is a photo of one of the famous baseball players who had ALS at a very early age. His name is Lou Gehrig's, and in the US, this disease is popularly called Lou Gehrig's disease. This is a very unfortunate disease. Even though it is considered a rarer disease compared to something like Alzheimer's or Parkinson's, the fatality or the life expectancy being very short after clinical diagnosis, and it increases the chances of aging population getting this disease so much so that it is expected that 1 in every 800 people are going to, end up with ALS with the growing aging population. And the unfortunate thing about this disease is its etiology, which is 90% sporadic, unknown causes, whereas only 10% have been familial, related to familial causes. And worse thing about this disease is the symptoms include muscle weakness and atrophy, and things like hyperreflexia, which is extremely debilitating, and it is a high cost for both the patients and the caregivers to live with this disease, to serve people suffering from this disease, to see them that way. And unfortunately, there is absolutely no cure, and even any kind of symptom management therapies that are out there are very minimally effective. So there is an immediate need for us to develop rehabilitative strategies, of course, therapeutic strategies, and also the reason for failure of these therapeutic strategies is because we know so little about what exactly is going on and where are the therapeutic targets and when is the therapeutic window. And this relates back to the lack of objective biomarkers and strategies to diagnose this disease early enough. So we kind of are interested in that area in the big picture. So just to give you a prelude to the pathophysiology of ALS, many of you may be familiar. This is also known as the motor neuron disease because the primary pathology is the death of the motor neurons. And motor neurons, as we know, are the neurons that integrate all of the nervous system inputs, and they excite or they drive the skeletal musculature, and therefore they're important for all kinds of all voluntary movement that we can think of. And in ALS, one of the known pathology is the retraction of these neuromuscular terminals, after which the muscles start to atrophy. And the reason for retraction is basically the fact that the motor neurons are degenerating. So this is just a simple cartoon showing you a healthy motor neuron versus an ALS motor neuron, and you can appreciate this retraction of the axonal terminals of the motor neurons from the muscle. So things are not simple, as we all know. So in the motor system, there is diversity, there's a lot of heterogeneity. Even within the motor neurons that are innervating the same muscle, we know that there is different subtypes of motor units, basically the combination of the motor neuron with its muscle fibers. So we have the fast fatigable motor units, the fast fatigue resistant ones, and the slow ones. And the reason for mentioning this and highlighting this is because of the fact that the disease ALS affects each of these motor neurons differentially. In other words, there is selective vulnerability of a subtype of motor neurons compared to the other ones. And these results are primarily driven by the studies in the mouse models of ALS, especially the SOD1 mouse model, which has been around for a long time now and has been widely studied. And all of the work that I will be presenting is also relating to this mouse model. So what is happening in ALS? At least at the peripheral end, we know that from the beginning of the birth all the way till the end of the disease progresses, and even before the so-called symptoms onset, there are changes happening. And one of the biggest changes that have been observed is the retraction of these fast fatigable motor unit innervation. So first, these FF units are gone, and it is also known based on mouse studies that there is some amount of re-innervation by the residual, let's say S type of motor units, the slow ones. And that's about when the weakness onset happens and the clinical symptoms sort of begin and eventually ends up in paralysis and the death of the animal. I just wanted to bring a really beautiful set of work from a group in Switzerland. This is basically highlighting the fact that the use of the animal models have allowed us to investigate more comprehensively the cellular and the molecular basis of the disease during the pre-symptomatic phases. So here is an example of the retraction of or the loss of the intramuscular axonal nerve, which is present here in the wild type animal versus which is almost completely gone in this mutant mouse on the right side here, right panel here. And note that this is at a pre-symptomatic age, which is the postnatal day 55. And typically these mice develop symptoms at about P90 and later. So this is early change even before we can see significant so-called clinical symptoms, clinical motivated symptoms. So in these mice at this stage, I also want to highlight how the difference exists in the fast twitch motor muscle versus the slow twitch muscle degeneration. So this is the LGC, the lateral gastrocnemic muscle, versus the soleus, which is the slow. So there's the fast and the slow. And you can appreciate here, as the age progresses, the axonal or the intramuscular nerve and the innervated neuromuscular junctions, they drop dramatically at about P50 here. In fact, the neuromuscular junctions are lost ahead of the axonal nerve loss. And this is really dramatic in the fast twitch muscle, whereas in the slow one, as you can see, it is much more preserved. And the loss of the axonal nerve, in fact, happens at about an age where the so-called clinical symptoms begin. So this is a beautiful summary of the various types of changes that occur both systemic level, the spinal cord level, and more specifically, the cellular level on the motor unit level. The take home from this is really the idea that the disease is progressive, changes are happening pre-symptomatically, and there is selective vulnerability of certain motor units versus the other. And also notice the variety of mechanisms other than, within the central nervous system. For example, the microgliosis, astrogliosis, so the involvement of glia is becoming increasingly recognized. And at the cellular level, there are lots of stress responses. There is mitochondrial dysfunction, there is the endoplasmic reticular dysfunction, and a lot of problems with the protein machinery. So there is protein dyshomeostasis and a lot of issues there. But this was a review from back in about 10 years ago. So we knew these things. What really motivated our work at that point was this idea that the abnormalities exist, the earliest ones being in the electrophysiological properties of the motor neurons. So more specifically, we are interested in the bulbar system, which is essentially the motor neuron, which consists of the motor neurons in the brainstem area, which control all the oropharyngeal muscles. And bulbar symptoms are very, they are pretty predominant in ALS. About 1/3 of the ALS cases turn out to be bulbar onset ALS. And you can appreciate that there are other ways in which ALS begins. It manifests itself. So there is the spinal onset, there is the primary lateral sclerosis, progressive muscular atrophy, and the bulbar onset. What is shown here in this recent review in the New England Journal of Medicine is highlighting the fact that the bulbar onset is really dramatic in terms of the shortness or life expectancy being very short. So the patients who have the bulbar onset ALS live very, less number of years as even compared to the others. And overall, the life expectancy in ALS itself is so short after clinical diagnosis. So we were interested in this, understanding these bulbar motor neurons, what is happening with them. So we focused on the jaw circuitry, and studies have shown that in the mouse model as well, the SOD1 mouse model that I'll be talking about, there is a lot of changes happening at the pre-disease level itself. So as noticed here, there is a drop in the rate of licking, rate of mastication, and together this leads to the animal consuming possibly less food and also losing body weight. So initially in the pre-disease, there is the body weight seems to be somewhat reasonably similar to the wild type, whereas later on, as you can see, there's a dramatic drop. But what is crucial is the causative factor could be that these pre-disease changes or the pre-symptomatic changes that we see here. So my postdoctoral work at UCLA in Dr. Scott Chandler's laboratory is when I started looking at this disease and the various things that we have been doing. This is where my work on ALS began. So I looked at the early electrical properties which were disturbed in the motor units of the controlling the jaw movement. So we were interested in the jaw motor neurons, and we looked at a very early stage, early time point, which is definitely pre-symptomatic. The mice are still in their postnatal day 10 plus or minus 2 days. And our goal was to test to see if there is the selective vulnerability that I just talked about in the fast versus the slow. Does that manifest at the very beginning itself? So we went ahead and we located the specific motor units. So we targeted the jaw closer motor pools and recognized these motor neurons using a retrograde tracer in the brainstem. And I conducted electrophysiology in these motor neurons. And our results are, this is a very quick summary of what we found. This is just showing our localization approach of the motor neuron and the recorded motor neuron here. But on the right-hand side here is a primary result that I would like to share with you. So what was, again, interesting and consistent with what was known at the periphery, which is the loss of the fast fatigable motor units first, but that is even at a later stage, about P55 that we saw. But even as early as P10, we were able to distinguish, of course, first of all, we recognized these motor units by using an indirect methodology based on the electrophysiological properties of these neurons. We segregated them into the 3 different types using a clustering approach. And this is shown to be at least 95 to 97% accurate in predicting the right type of motor unit or classifying them. So we used this approach, and we then found that only the FF, the fast fatigable and the fast fatigue resistant motor units that we recognized through such a classification approach were the ones that were affected. And they seem to have a reduced input threshold to eliciting action potentials, meaning they were having this hyper excitable shift. So they were becoming somewhat hyper excitable very early on. And this reduction in the input threshold is very crucial for motor neurons, especially as we all know, because recruitment of the motor neurons is an important property to elicit appropriate muscle responses. So I kind of got interested in trying to see if we can predict what could happen to the muscle force, given these different types of changes that we observed in the data, the lower recruitment threshold, the dysregulated input-output relationships, such as the injected current and the spike frequency here. As you can see, the wild type, the 3 different types of units that we identify nicely line up as expected, showing a nice recruitment order here, whereas in the mutant, it was all kind of muddled up. So it was a very curious question as to what could happen to the force if this was the nature of the motor unit distribution. And also there was quite a bit of reorganization of the motor pools itself in terms of the distribution of these motor units. So together, I kind of put them all together and used a frequency force transformation and predicted this type of a force change in the so-called isometric force. So this is just an input current that is typically applied to these motor neurons. And the black line here is the expected control behavior based on the properties of these wild type data that we had. Whereas the modified, the SOD1 properties, when we incorporated that, we saw that the force curve became rather steep. Of course, there is no evidence that we know of that the amount of force itself declines at this stage. Nonetheless, given these properties of the neurons, we saw that there was a drop in the dynamic range of the force generated in the SOD1. So this is yet to be behaviorally tested. These are not easy things to test because of the lack of reliability of many of the techniques that are available. But this just prompts us to pursue such more precise techniques to begin measuring some of these things early on, such that this will hopefully eventually have some translational value for early disease prediction. So adding to the diversity of phenomenon that's happening in ALS, it is also known that most of the motor pools or the motor neurons are dying in this disease. However, there are some motor neurons that remain spared. For example, the oculomotor neurons that control the eye musculature and also the motor neurons involved in bladder control. These remain, they remain resistant to the disease compared to the other motor pools throughout the brain and the spinal cord. So we were interested because the two nuclei that I'm showing here are closely situated. It is possible to say, okay, so let's look at both of them from the same mouse and see and compare what happens to the early electrophysiology. So I collaborated with a research scientist in the lab at that time, and I mentored a undergraduate student, Takuma Sonoda. He's now gone on to Northwestern, and he's actually he got his PhD and published a paper in Science as the first author, discovering new types of cells in the retina, actually. So I'm very proud to share that story. Now back to the motor neuron story here. So the oculomotor neurons that control the eye musculature, interestingly, they remained resistant to the changes in the electrophysiological properties as well. So this kind of suggested to us that the physiology of motor neurons is a key indicator and possibly, hopefully, also a modulator of the disease, which is yet to be proved. But we think that at least it reports the disease state quite well. So this was an interesting result that we observed. Now, again, going back to the same motor pool and the diversity of motor neurons, the three different types of motor units that I described earlier, the FF, FR, and the S types, they're all the alpha motor neurons. In addition to that, there are these gamma motor neurons that innervate the intrafusal fibers, basically the muscle spindles, which are the stretch receptors within the muscle, sensing the length and the velocity information of the muscle stretch. So it turns out that these gamma motor neurons, as shown by this beautiful study here published in PNAS recently, they showed that the gamma motor neurons also remain resistant to degeneration within the same motor pool. As shown in this figure here, which summarizes in the wild type, you can see that the alpha motor neuron numbers are decreasing with age in general, but in the disease, this is much more dramatic. So there's a huge drop in the number of alpha motor neurons that they counted, whereas the gamma motor neurons remain consistently stable. The numbers remain consistently stable. So this indicated that there is some sort of a differentiation or differential vulnerability based on possibly the way they are configured, the basic anatomy of this system. So one of the things that is common between the oculomotor neurons and the gamma motor neurons is that both of these types of motor neurons lack muscle spindle inputs, which was very interesting because this same study also showed that when they cut off the proprioceptive inputs to the motor neurons, they were able to keep them survive longer. The disease got delayed a little bit. So this kind of told us that these spindle inputs could be a source of vulnerability in the system. In other words, it makes sense because the spindle inputs are providing this ongoing sensory feedback all the time, even when the muscle is resting. So there is the sensory feedback, and this sensory feedback from a neurotransmitter perspective, it is a glutamatergic input. So there is constant bombarding of glutamate onto these motor neurons, and possibly that could be one of the reasons why this sensitivity to this spindle inputs exist. So because of that, our jaw system is turned out to be very unique. Now, this is just a cartoon showing the typical muscle stretch reflex. This is just a knee jerk reflex. And the basic thing I want to highlight here is this afferent information carried through these sensory neurons, 1A primary afferents, and then that relate to the motor neuron. So this is the reflex, monosynaptic reflex arc. And now here is what is the architecture in the jaw system, which is very unique. And this is one of the key reasons why we want to also pursue the jaw system so closely. So in the jaw system, there is also these jaw jerk reflex, which involves the sensory, monosynaptic sensory and motor circuitry here. But the interesting thing is that the 1A stretch proprioceptive sensory neurons receiving the stretch information from the muscles, they are located in the brain itself. Unlike any other system where these proprioceptive neurons exist in the peripheral ganglia, and they are kind of embedded amongst the other types of sensory neurons, the mechanosensory, nociceptive, etc. So the segregation and the isolation of these proprioceptive sensory neurons within the brain in this trigeminal system, the jaw control system, makes it a really unique target to study whether there is really the differentiable circuit-specific vulnerability in ALS. So this is just another cartoon highlighting the location of the different sensory neurons within the trigeminal system. The mesencephalic nucleus here, distributed along here within the brain, consists of the large majority of the proprioceptive sensory neurons, the 1A afferent neurons, carrying the stretch sensation. Whereas the other mechanosensory and the nociceptive, all of the other sensory neurons are located in the peripheral trigeminal ganglion here. So this anatomical segregation of the functionally different sensory neurons makes the system a really attractive one to study. So when I began my work as an assistant professor at UCLA, my postdoc, Soju Seiki, he pursued examination of the sensory neurons within the mesencephalic 5 nucleus, the trigeminal mesencephalic nucleus. And his work basically identified some of the abnormalities that we saw even at P10 when the motor abnormality was also found. So within the MESV nucleus, there are at least 2 types of discharge patterns that we see. One is a really nice rhythmic bursting. The other one is a tonic spiking pattern, as shown here. And in the SOD1 mouse that we studied, it turns out that there was a large majority of burstiness in these SOD1 neurons, as you can appreciate here. And not only that, these bursting neurons ended up being very arrhythmic, very irregular. So the bursting was very irregular in these neurons. And interestingly, when we compared this with the other trigeminal sensory neurons, the ganglion neurons, as well as the peripheral visual sensory neurons, both of those were devoid of excitability changes that we observed in these MESV neurons. So clearly there is some sort of a intrinsic abnormality within the circuitry itself besides the motor neurons. So this circuit-specific abnormality is very interesting, and it's kind of propelling our future directions because now we have a microcircuit in which we have seen these specific abnormalities very early on. And this kind of tells us this is a target to both track the disease progression as well as possibly to modulate the disease. So we got interested in what is causing this abnormality in these sensory neurons. And in the previous work, it has been shown that the sodium currents, the NAV 1.6 type of sodium currents, are extensively present in these neurons, and they control their excitability. So so we got interested in identifying whether these sodium currents were abnormal in these neurons. And what I highlight here, this is an example of the sodium current that looks like this. It has a fast component, it has a persistent component which exists for a long time, and also it has this unique resurgent component. So basically, this type of a current expression experimentally is done when we depolarize the neuron to a very high voltage, which generates this fast current, and then we repolarize it to a negative 40 millivolt sort of voltage, which generates this resurgent, and then we also see this persistent component. So I'll tell you more about this resurgent, why I'm so excited about that in a second. But what Soju saw in his data was that compared to the wild type, the SOD1 had much lower amount of both this persistent and the resurgent sodium currents. So this sort of explained the reduced excitability of these neurons that he saw. But we were also interested to see if this also explains the irregularity that we saw. So this is one of our undergraduates, Kiera Cohen. She graduated. She's now a graduate student at Johns Hopkins. And she, this is her data showing the protein expression as well in terms of the NAV 1.6 ion channel protein that is also reduced, consistent with our functional current expression here. So together, this told us this story that the sensory neurons are abnormal early on at about the same time where we see the motor abnormality. But thinking about the direction of change in the sensory, now the sensory is becoming aberrant, arrhythmic, and reduced excitability. So these are glutamatergic neurons feeding or supplying the excitation, ongoing excitation to the subjacent motor neurons, which are at this point somewhat hyperexcitable. So it's kind of counterintuitive to a popular sort of theory where they say excitotoxicity. There's this constant glutamate bombarding. Anyhow, before I go into the what we derive out of these studies, I now want to turn to some of the interesting components of our study, which I think many of you will sort of enjoy and appreciate. So continuing our story about this abnormal arrhythmic firing patterns of these sensory neurons and the connection with the sodium channels, we kind of pursued a study where we wanted to see if we can restore these sodium currents and see if the abnormality gets fixed. So we tried doing this at a cellular level. So we used this closed loop control of neural excitability. This is a technique called dynamic clamp. Before that, as a step one, we generated a novel computational model for this new type of new type of conductance, the resurgent conductance. So this type of NAV 1.6 sodium channel has a very unique operational characteristic. So the way this works is when the channel opens due to depolarization, there is a blocking particle that blocks this open channel. So you can imagine this to be, someone knocking at the door and you opening the door, but you're there blocking the incoming person from entering the house as the blocker. Now, this blocking has to be removed in order for sodium to come in. So removal of that blocking particle is called unblocking, and that unblocking allows the sodium to come in, and then the inactivation proceeds as expected. So this unique blocking, unblocking mechanism is what gives rise to this resurgent current. So we developed a computational model for this unique type of conductance, very unconventional conductance. And what we did was we used this closed loop approach, which basically involves putting your model in the computer here, and you're patching a neuron here. So you're doing your electrophysiology experiments here, and you're recording the voltage. And that voltage through the dynamic clamping is fed to your model neuron, model channel. and that updates the model current, and that model current in real time is fed back to the neuron in question. And this forms the closed loop control of the neural excitability by making this model current as an in silico knock-in of a channel. So by doing that, and by developing a model that segregates the 3 different types of sodium currents, we were able to test their role in control of neural excitability, and also whether restoring those components further restore the rhythmic bursting in these neurons. So this is a really nice trace showing the neuronal burst discharge in a bursting mesencephalic sensory neuron. And addition of this newly modeled resurgent current showed the increase in the burst duration as well as the interburst interval, which was also predicted by a computational model of this sensory neuron. So for this work, I developed the model, and I collaborated with Dr. Antonio Spantasis in the medical school, really talented scientist who helped us with this dynamic clamp, getting it to work. And we conducted these experiments in their lab. And also Dr. Soju Seki, who conducted these actual dynamic clamp experiments are here. And so next, Soju was interested in figuring out if this approach would help us restore the normal or rhythmic bursting patterns in the SOD1 abnormal sensory neuron. So what is interesting about such an approach is that this in silico knock-in, it avoids all of the side effects of the pharmacological approaches. So it's kind of a cleaner way to test theories and hypotheses about the roles of ion channels. So back to here. So here is a burst discharge in a SOD1 MESV neuron with these irregular interburst intervals. And when we enhanced the in silico sodium currents, we saw this more rhythmic type of burst discharge. And when we removed it, the irregularity came back. And this rhythmicity was comparable to the wild type sensory neuron. So this was really interesting to us. And again, then I collaborated with Dr. David Turman at Ohio State to kind of do more deeper mathematical analysis of what actually these changes in these ionic currents do to distort the stability of the neuronal discharge. So this study was solely focused on identifying the role of these resurgent current. We already know persistent current is important for bursting. It causes bursting in these neurons along with other potassium currents, but the resurgent was not clear. So as you see here, even without resurgent, the model neuron here, it can burst. But when we add noise and make it noisy, like we saw in the SOD1, when we add the resurgent current to the model, it fixes that noisy behavior. I have this really beautiful figure, which I admire a lot. It's one of my favorites, but I don't want to bore you with all this abstraction. But if you do recognize yourself with these types of figures, please feel free to talk to me later, and I'm happy to answer your questions. But together, the overall take home of this analysis is segregation of the different roles that these persistent and resurgent currents are doing to the cell. And what we found was that this cell, which has this property of resonance, basically it is selective to certain input frequencies that causes the resonance property, which is dependent on this persistent sodium conductance. It causes the neuron to tune in to expected input frequencies. However, it is not capable of segregating the noise from the noisiness, which may arise intrinsically, extrinsically. We don't know the source of noise or the type of noise. In any event, it is not robust to the uncertainties that could be present. With the resurgent current, it is able to tune out that noise and exhibit reliable bursting. So this kind of told us that not only are these currents playing crucial roles in the sensory coding of these neurons, but in the disease, this is getting disrupted. So information is getting disrupted early in the disease. So if information does get disrupted, what could be resulting from that kind of an interaction at the circuit level? So I developed this very simple sensory motor monosynaptic reflex circuit type of model. And I mimicked the sensory irregularity, and correspondingly, together with some of the changes that we observed in the motor neuron, such as the hyperexcitability, when we incorporated that, what we saw was that the motor neuron, sometimes it liked to be synchronous. The activity of the motor neuron was synchronous with the sensory. Of course, this motor neuron has plateau potentials, which makes it, fire longer, as you see here. But occasionally, when the sensory neuron was doing its mixed up behavior with these short distance between bursts and such, the motor neuron, on the other hand, together with its plateau property and this short burst interval of the sensory, it fused its spikes together. So how is this really relevant to the disease? I mean, what does it have to do with the disease? So to be completely honest, at this point at P10, we still don't know exactly what happens to the muscle output if this is the sensory motor circuit. However, it is known that in ALS, the early symptoms are muscle fasciculations. They may or may not be troublesome. It may not be painful or anything of that sort, but they do occur very often, and that's a very first symptom in ALS. So this is showing a first symptoms of ALS, this early polyphasic, unstable fasciculation potentials in an ALS patient. And I'm showing you these 3 bursts. It actually had 8 continuous consecutive bursts. So this type of activity in the motor neuron and the biophysical basis here that we show could be a factor that is contributing to eventually causing such fasciculations in the system. So going further forward, what we want to do, of course, we want to look at the circuitry, the synaptic interfaces, as well as the information change that occurs because of the changes in the physiology and the biology underlying that. But we also want to link the molecular changes to what is happening at a circuit level as well as at the behavioral level, because much of the disease research is largely focused on identifying the transcriptomic changes, proteomic changes, and things like that. So it would be of value to kind of link them all together to really appreciate the total outcome of what the disease does and how that changes the behavior. So we collaborated with Dr. Yang's lab in our department along with Dr. Chandler's lab, and we did single cell RNA sequencing in these brainstem neurons. This is at a disease stage, in other words, a symptomatic stage. This is showing the existence of different types of cells in this. So the primary finding from this study is really highlighting the fact that it is not, again, just the motor neurons that are suffering. It is a whole cohort of different types of cells, especially the glial cells, which are definitely participants in these changes that are observed in the disease. And exactly how they contribute and exactly how they manipulate the circuits is still unclear. And that's a huge open-ended question that we have. To summarize, there are these various mechanisms that are known to be involved in ALS. And some of the prevailing theories are kind of summarized nicely in this beautiful figure here from a Nature review. So what is really shown here is it's a busy figure. It's a lot of stuff going on. But the big idea is there is one of the first theories that emerged out of the motor neuron intrinsic property measurements was the idea that there is excitotoxicity. So there is glutamate, which is uncontrollable in the synaptic clefts, and that glutamate is bombarding the motor neuron with a lot of excitation, and that is in turn bringing in a lot of calcium, and there is calcium dyshomeostasis, mitochondrial and ER imbalance leading to motor neuron death and degeneration. So that was the sort of the early hypothesis that came about with all the studies. But now we know that the glial cells are heavily involved, the astrocytes, the microglia, the oligodendrocytes, and also the axonal deficits. But most importantly, the protein machinery. The protein quality control is very poor, it is disturbed, and also the RNA metabolism is messed up. So together, there are all of these different things going on. So what do we think, what is going on? So from our work, it is clear that early on, there is the intrinsic change and the circuit change. But how do we recognize this change? Is it disease or is it compensation? Because early in the disease process, there is no symptom to call it necessarily the disease. So how do you perceive these, and how are these different mechanisms linked with one another? So this is kind of how we perceive it. The early changes that we see perhaps is still compensation. The increase in excitability of motor neurons could be a response to a decrease in the sensory excitability. So that also involves circuit changes, and we don't know exactly why this circuit change is happening. But we have some evidence, which I did not show you, that there are these changes in the inhibitory end as well. So there is the GABAergic change. So there is excitation, inhibition, imbalance, or imbalance to compensate for the intrinsic changes. So these two possibly are balancing one another. And of course, each of these compensation relies on changes in different protein expression levels and so on, which then keeps this dial of the protein homeostasis. So this interaction, we think, might be responsible to keep this balance. So what is disease doing here? Well, we still don't know, but this is what we think. It is likely that the disease, it messes up the calibration of the system. So by pushing the calibration to one side or another, it is kind of misleading. It is elusively, misleading the whole system to remain connected in the wrong way. So what is important here is there is a lot of work to be done. We still don't know so many things about what is going on. So the circuit approach together with the molecular approach is absolutely essential. But also essential are computational approaches, which help us link these different components, different types of changes. And as we all know, these types of changes cannot be measured with one approach. So there has to be multiple experimental approaches, and the computational work has to link all of that and develop a model for us to test how disease actually perturbs the function of the system, the molecular underpinnings of the disease, and the circuit operation. So looking ahead, we want to continue this work with experimental approaches, which will help us identify and model the functional properties of proteins as well as the composition of these proteins. And we are interested in the heterogeneity of the genetic basis of ALS. So we are looking at multiple mouse models using diverse experimental approaches. We want to use the computational modeling approach and the simulations and the theory that help us decipher more about the intrinsic workings of these things. And also closed loop interfaces will be of great importance, not just at the cellular level, but also at the organismal level and at every level, in fact. This will really help us test the hypothesis and also develop strategies to control the disease, definitely. And that's a big one for us. And we're interested in microcircuitry, and we have the tools and the anatomy that tells us the disaggregation of the different cell types. So that way we can model the disease in these different types of cells and its progression as age goes on, and test our big hypothesis about disease development. I would like to conclude by thanking my collaborators, Dr. Jia Yang and Dr. Scott Chandler in our department, and Dr. Ricardo Olchisi and Dr. Martina Vito. They are my collaborators in the medical school, and Dr. Igor Spiegelman, whose lab helped us with the trigeminal ganglia work in the dental school, and of course, my grant support from NINDS. Thank you all for listening, and I'm ready to take questions if you have any.
Thank you. That was nice. I'm sure everybody is giving you a virtual round of applause and
thank you. Thank you very much.
In our seats. Yeah, so there are perhaps a couple of questions that I've been seeing appearing. Maybe I'll read a couple of those. So Paulette asks, the modeling of the NAV 1.6 channel sounds really interesting. The blocker in the NAV 1.6 channel, is that an in silico parameterization, or does that have any physiological representation? I guess
yeah. So the blocker does have a physiological manifestation. There is a known particle, that cytosolic particle, that people, one group in Northwestern, they have done most extensive work in this area, and they have found that there is indeed a beta particle that actually forms a putative blocker. So it is a cytosolic molecule, and when the channel opens, they showed that by manipulating this beta particle, they could show that they could manipulate the blocking function of this channel. So it does exist. And in our model, the resurgent current is a very complicated, it actually was quite interesting to model it. It was not one of those typical currents like the sodium or the potassium that we know. So we kind of had to do like a hybrid model of that, which involved like a state change kind of approach, as well as this nonlinear behavior of these channels. So the model that we have developed is it's a conductance-based model for this current. I hope that answers your question.
Yeah, I think that was a thorough explanation. I've noticed a couple of questions coming in chat. So one from Camilla was there reduced jaw muscle function in your animal model as early as P10?
Yes, that's a great question, and we still haven't measured it. We have to devise, so there is the basic problem is the technique. So we have to have a reliable technique that actually helps us, tell how exactly would you measure it reliably, because these are very small animals, and they are very susceptible. I mean, if you touch them too hard, they will die. So we have to be super careful. So there's some, these technical limitations that are keeping us from trying something, but we want to do that. And we hope that maybe something like looking at their feeding behavior, the licking rate, things like that, those kinds of measurements might be possible. So we are thinking about those things.
Great, excellent. From Professor Raj, we have a question asking about the details of the technical details of your dynamic patch clamp, for example, what interfaces you're using, what electrodes you're using. And he's wondering what advances you would like to see on the electrode technology. And just for a bit of background, Professor Raj is in electronics packaging and biocompatible electronic systems.
I see. Very good. So my experience with the dynamic clamp, it was a really interesting experience. And what we used was the traditional RTXI system. So the electrodes were the same kind of patch electrodes. There was no difference in that aspect of that project. It's mainly the dynamic clamp system that integrated the patch clamp amplifier to the cell. Basically, that's where the interface came. What I would like to see is, the ease of use of that system. So typically, when electrophysiologists try to adopt such systems that require more programming and more sort of technical manipulation of the models, first of all, saying, it's a model, it's a dynamic model, go ahead and install that, itself is somewhat intimidating to many of the experimental community. But now it's not seeming to be the same. People are adapting to using programming and modeling and things like that. They want to do it, and they see the value to it. So I think if the system is adaptable as easily as the patch clamp amplifier itself for an experimentalist, I think that would be less knobs to tweak and easy to sort of connect and adapt. Calibration and all of those things, if it is straightforward, I think that will be a great start. Yeah.
Wow, Professor Raj, you got your work cut out for you building plug and play biocompatible packaged electrodes. Okay, maybe I'll ask one of my own here. When you were talking about the recruitment patterns of, I think it was motor neurons becoming muddled through the disease progression, that reminded me a lot of what many of the neural engineers in our department face when they want to do things like stimulate motor neurons to activate muscles, where you pick up the wrong recruitment patterns. So do you have any idea if there's an analogy between these? I mean, is the muddled recruitment patterns substantially affecting the force and dexterous manipulation that's available for the muscles that they innervate?
I hope I understood your question correctly. Correct me if I'm wrong. So at least in the model that I showed you, it was a very simplistic model. So we do assume an orderly recruitment, and the experimental realism is built into the model based on the number of cells that we observe, and we arrange them in the recruitment order manner, and we kind of incorporated their frequency current gains into the model. So I would say it is a pretty reasonable replication of what the motor pool composition would be. And when we did the same thing with the SOD1, that came out different. But as you could see, the sigmoidal curve, the shape was pretty similar in both cases. What really changed was the dynamic range, which tells us that there is some value to this prediction, which the only caveat right now is that we haven't experimentally tested it. So that kind of testing, as one of you already brought up, can we test this in the muscle? And I really would love to see that. And that's what we're hoping to see with the behavioral tests that are coming up. Yeah.
Excellent. Well, thank you. I think that covered the questions that I noticed, and we're basically right on time too. So thank
Sounds
you again
great.
for your talk. This is great. Really appreciate it.
Sounds good. Thank you.